In the beginning I mentioned that the amide-1 band is shaped by hydrogen bonding and the coupling between local amide-1 vibrations. The latter makes that the amide-1 absorption is sensitive to more structural features than just secondary structure. This is discussed in the following. There is no point for you to be able to reiterate all these structural influences in detail, but you should appreciate the sensitivity of coupled vibrations to a number of structural features. For example, the amide-1 spectrum is sensitive not only to the secondary structure per se, but also to the assembly of secondary structure elements. This is shown here for alpha helices with the help of calculated spectra. The formation of alpha helix bundles causes an upshift of the amide-1 band because vibrations in adjacent helices couple with each other. The left figure shows the situation when two helices are brought together. The peak wave number is higher when the helices are close than when they are far apart. At very large distances, the helices are uncoupled, which case is shown with the blue spectrum. The right figure illustrates the effect of assembling several helices. The more helices are contained in a helix bundle, the higher the wave number, as shown here for 2 helices, 4 and 7 helices. As you know, helix bundles are common structures in membrane proteins, so this shift is a relevant property of the amide-1 vibrations. The helix bundle effect is another example for the structural sensitivity of coupled vibrations. The next example are beta sheets. In the discussion of tender-mistat denaturation, I mentioned already that intermolecular beta sheets of protein aggregates absorb at lower wave numbers than the intramolecular beta sheets of globular proteins. The explanation for this observation is that the intermolecular beta sheets tend to have more strands and more planarity. The strand effect is illustrated in the figure, which shows calculated beta sheet spectra. We can focus just on the antiparallel sheets, which are associated with the blue spectra. Their main absorption band shifts down when strands are added to the sheet, as shown here for structures with 1 strand, 2 strands and 4 strands. A larger number of strands and more planarity enable more and stronger coupling of the amide-1 vibrations. Thus, the infrared spectrum does not only indicate whether the structure is a beta sheet or not, but is also sensitive to additional structural details. And this is just another example for the structural sensitivity of coupled vibrations. The size effect inferred from spectrum calculations can also be seen in experiments. The example here are our studies of the aggregation of the amyloid beta peptide, which is important in Alzheimer's disease and may be one of its causes. The slide presents second derivative spectra of several amyloid beta oligomers. The main beta sheet band of these can be seen near 1630 reciprocal centimeters. The bottom spectra are for 2 oligomers prepared in detergent. They are relatively stable over time. The red spectrum is for a preparation of small oligomers that consist of approximately 4 peptide molecules. The oligomers associated with the blue spectrum are larger and consist of approximately 12 peptides. It is clear that the larger oligomers absorb at lower wave number. The other spectra were obtained in a time-resolved experiment in the absence of detergent. The initial spectrum is the black spectrum on the top of the figure. Its amide-1 band is found at even lower wave number than that of the dodecamer. It shifts further down with time as shown in the middle spectra, from the blue spectrum to the red spectrum. After 20 hours, the oligomers consist of approximately 30 peptide molecules. In addition to the shift, the band becomes narrower with time on its high wave number side, as you can see here, because there is less structural heterogeneity at later times. Thus, amyloid beta oligomers of different sizes exhibit their main beta sheet band at different wave numbers. The larger the oligomer, the lower the band position. In kinetic experiments, the band shifts down with time and becomes narrower. This indicates that the population of beta sheet structures becomes less heterogeneous with time. And this experiment is another example for the structural sensitivity of coupled vibrations, as well as for the sensitivity of the bandwidth to structural heterogeneity. When beta sheets assemble in beta sheet layers, there is also a shift of the amide 1 absorption to higher wave numbers. The figure shows the effect when two beta sheets are brought together and form a beta sheet layer. The uncoupled case is shown in black, and the closer the two sheets are, the higher is the wave number. The effect is caused by the coupling of vibrations in different sheets. Such beta sheet layers are formed in amyloid fibers, which are involved in a number of diseases. Now it starts getting boring, but this is another example for the structural sensitivity of coupled vibrations. Finally, I want to show you that 13C labeling is a very powerful approach thanks to vibrational coupling and thanks to the mass effect. This is shown in the following slides. The bottom spectra in this slide are absorbance spectra, and the top spectra second derivative spectra. The blue spectra are those of the unlabeled amyloid beta oligomers, while the red spectra are obtained with the 13C isotope. 13C labeling shifts the entire amide 1 spectrum down by about 40 reciprocal centimeters because of the heavier mass of the 13C isotope compared to the ordinary 12C isotope. One of the obvious advantages of the isotope shift is the possibility to analyze the structure of both the labeled and the unlabeled protein or peptide in the same experiment. This can be used to study the interaction between two proteins or peptides. When they form a complex, they might undergo conformational changes. When one of them is labeled, one can characterize the conformational change of each interaction partner. Another advantage is that 12C and 13C groups do not couple much with each other. As mentioned earlier, coupling is strongest when the local amide 1 vibrations have the same frequency. Because of the mass effect, the 13C groups vibrate slower and couple only very little with the 12C vibrations. Instead, they disrupt the vibrational coupling between the 12C groups. As a consequence, mixing of 12C and 13C peptides in beta sheets can be detected. It affects the position of both the 12C and the 13C band. We've used this, for example, to show that the two main variants of the amyloid beta peptide, with 40 and 42 residues, form common beta sheets where the two variants are randomly or nearly randomly mixed. When labeled and unlabeled versions of the same peptide are mixed, they form homo-oligomers. At low concentrations of the labeled peptide, the labeled peptide will be surrounded by unlabeled peptides, as shown in the figure. The 13C amide groups can then only couple with other 13C groups in the same peptide. The figure shows a beta sheet, where the strands of the single labeled peptides are shown in red. Here, here, and here. The blue strands are from the unlabeled peptides. On the top, the labeled peptide contributes only a single strand to the beta sheet. In the middle, it forms a hairpin, and on the bottom a 3-stranded sheet. There is more coupling between the 13C groups in the hairpin than in the single strand, and there is even more coupling in the 3-stranded sheet than in the hairpin. Therefore, vibrational coupling can be used to analyze the structure of individual peptide molecules within an oligomer. In this way, we could find out that each amyloid beta molecule contributes 2 or more adjacent strands to the beta sheets of the oligomers, which exploited the mass effect and the effect of vibrational coupling. More detailed structural information can be obtained when specific amide groups are 13C labeled. They do not couple when they are distant in the three-dimensional structure. When they are close, however, they couple, which leads to a change in the spectrum. On the left-hand side, the labeled sites are distant and there is no vibrational coupling between them. On the right-hand side, the labeled sites are close and there is intra- and intermolecular vibrational coupling between them, which is indicated by the arrows. This changes the spectrum and therefore it can be detected whether the 13C groups are close or not. In this way, closeness between specific amide groups can be detected, which is analogous to contact information from nuclear magnetic resonance spectroscopy. And by now you know which effects are responsible for the labeling effects. Summarizing this part, vibrational coupling makes the spectrum sensitive to many structural properties, like the number of strands and their twist in beta sheets. In addition, it makes the spectrum sensitive to the three-dimensional assembly of secondary structure elements. Vibrational coupling can be further exploited when combining it with the mass effect of 13C labeling. This provides further information on the structure. It enables to analyze the structure of individual mixture components, to detect strand mixing in beta sheets, and to detect contacts between specific groups.